So in this discussion, I want to spend some time talking about the interesting and unique properties of hemoglobin. Some of the things here I've talked about already in the lecture, but I sort of want to explain them in another way and hopefully help clarify points for you about the oxygen binding properties of hemoglobin. Now, hemoglobin, you may remember from the lecture, is a molecule, of course, that is related to the molecule myoglobin. Both have a very similar property in that they both are oxygen-binding proteins. Big difference between the oxygen-binding property of hemoglobin and that of myoglobin, as I mentioned in class, is that hemoglobin is very well set up to vary its oxygen binding properties as a result of the oxygen environment in which it finds itself, whereas myoglobin does not have those properties. Myoglobin tends to be very good at binding oxygen, but it only lets go of its oxygen at very, very low oxygen concentrations. And we can actually see that in this graph. So in this graph, we're looking at, on the y-axis, the percent saturation of each molecule for oxygen. What percent is it saturated? Up at the very top, of course, 100% saturated, down at the bottom, 0. And we're looking at that saturation as a function of the oxygen concentration in which it finds itself. Well, if we look at myoglobin, we see that even at fairly low oxygen concentrations down here, myoglobin is fairly well saturated with oxygen, meaning it's very good at holding on to it. Myoglobin is what I like to think about as an oxygen battery. It holds on to oxygen until oxygen is almost all gone. So our muscles have myoglobin in them so that when they really start running out of oxygen, when the oxygen concentration gets very, very low, myoglobin lets go of oxygen. But that's the only time it lets go. Otherwise, it's grabbing and holding on to oxygen to prepare for those conditions. Hemoglobin, on the other hand, varies in its oxygen binding properties. When we look at these graphs that I'm going to be showing you over this set of talks, as you see a curve shifted to the right in these oxygen binding curves, that means reduced affinity. That means it's letting go of oxygen better. So as we compare the oxygen binding curve for hemoglobin compared to that of myoglobin, we see that it has shifted to the right, and that means it has a reduced affinity for oxygen in general. And we can actually see that if we look here where we see what's called the P50s. That's the value, the concentration of oxygen necessary to get each of these molecules to 50% saturation. And you can see that the P50 over here for myoglobin is significantly different than the P50 value for hemoglobin. It takes a lot more oxygen for hemoglobin to be 50% saturated. Okay? That means, therefore, that hemoglobin is very good at letting go of oxygen. If we look way up at the top, we see high concentrations of oxygen, and up here at the top, what do we see? We see that both of these are saturated for oxygen. So at high oxygen concentrations, hemoglobin is just like myoglobin. It gets saturated very readily. Well, that happens in our lungs when we're breathing. We want hemoglobin to get saturated with oxygen because that's what it's doing. It's carrying oxygen out to the tissues. And as it gets out to the tissues, what happens to the oxygen concentration? Well, it starts falling. And what happens to the percent saturation of hemoglobin? It starts falling as well, meaning that at lower oxygen concentrations, hemoglobin is letting go of oxygen, whereas myoglobin is holding on to it pretty tightly. Now, the binding of oxygen that both myoglobin and hemoglobin do happen as a result of what's called the heme group. That's what gives hemoglobin its name. Both myoglobin and hemoglobin have a heme ring. Hemoglobin has 4 heme rings because it has 4 polypeptides. Okay? Myoglobin has 1 heme ring because it has 1 polypeptide. The heme rings themselves that you can see here are identical. The functional part of the heme ring for the binding of oxygen is shown right here, and that is, get my finger pointed right, that is the iron atom. The iron atom, iron in the +2 state, the ferrous state, is the one that binds to the oxygen molecule, and you can see that. And this is actually attempting to show you in 3 dimensions, in this sort of a weird drawing, how that oxygen binding occurs. Now, if we look at the very bottom here, we see this ring structure. This ring structure is actually the side chain of a molecule of histidine. This side chain of histidine is attached. Notice it's attached to the iron atom that you can see right here, okay? Now, that's important because, as we'll see in the next slide, the binding of oxygen actually does something really interesting with the iron atom. It pulls it up very slightly. And as I've said in the lecture, very tiny changes in protein structure change protein properties, in some cases, drastically. Well, in this case, this tiny change in the position of the iron atom that happens when oxygen binds causes the iron atom to move up. It also causes, as a result, the histidine to move up because it's attached to the iron. We can actually see that a little bit better here. So we see on the very top figure, we see deoxyhemoglobin. We're looking at the heme group from the side chain now instead of looking at it sort of angled as we were before. We can see the iron atom in the middle, and we see that it's pretty much in the plane of the heme, okay? That is, it's a fairly flat structure. When oxygen binds, as we see down here, what happens? Well, we see that oxygen is bound, and we see now that that iron atom has moved up slightly. As a result of the iron atom moving up slightly, the histidine moves up slightly. Well, the histidine is attached to all of the other amino acids in the protein, so a slight change in the position of the histidine means that all of those other amino acids also very slightly change position, and so as a result of this slight change, we've made a complete slight change in the position of all of the other amino acids in that polypeptide chain. Well, for myoglobin, that's not a big deal. Myoglobin only has one polypeptide chain. If you change its structure, you change its structure, but nothing much else really happens. Hemoglobin, on the other hand, has 4 polypeptide chains, each polypeptide chain having one heme group. When we change the structure of one polypeptide chain by binding oxygen, this happens in the lungs when the hemoglobin comes into the lungs, for example, that first polypeptide chain changes its configuration. And when it changes its configuration, it changes its interaction with the other 3 polypeptide chains that are there. Now, what actually happens is that change of interaction between this heme group in this polypeptide chain with the other polypeptide chains means that the other polypeptide chains now have what we call greater affinity for oxygen. They want to bind oxygen more than they did before this happened in this polypeptide chain. The affinity of those polypeptide chains has changed as a result of binding of oxygen, and this turns out to be really important because now those polypeptide chains are ready to bind oxygen, and they're ready to bind oxygen quickly. Why does that matter? Well, the hemoglobin's in the lungs for a short period of time. We want to get it loaded up with oxygen as much as possible. When oxygen binds to one of the 4 chains, it favors the binding of oxygen by the other 4 chains. Okay? What we've done is converted hemoglobin from a state where it didn't bind much oxygen at all, that's before the first one bound, to a place where it wants to bind more after the first one has bound. What we've done is we've changed the overall structure and affinity of hemoglobin. We've changed it from what's called the T state, which it stands for tight, meaning it doesn't want to bind oxygen, to the R state, meaning relaxed, where it's happy to bind oxygen. And that happened because that very first oxygen bound. That phenomenon that I've just described to you is something that's known as cooperativity. Binding of one oxygen favors the binding of additional oxygens. Now, if we go back and look at the figure that we had before, we see the myoglobin binding and the hemoglobin binding, and now we begin to see why that oxygen binding affinity changes. And in fact, if we look at this, we can see that myoglobin had a figure that had a shape we called hyperbolic. Hyperbolic curves, okay, look like this. And you can see there's pretty linear, and then as the molecule starts to get saturated, it just starts bending over like that. There's nothing particularly interesting about that. On the other hand, hemoglobin has a curve that we call sigmoidal. It comes out and it forms an S-like structure, and that's reflecting the fact that the affinity for oxygen is changing. The more oxygen we put into hemoglobin, the more it wants to change and the more oxygen it wants to bind. Well, hemoglobin has some other very interesting properties. Now, I've told you, of course, when it gets in low oxygen concentrations, it wants to let go of oxygen. And that makes sense. We've got high oxygen concentrations in our lungs. We have lower concentration of oxygen out in our tissues. And so when hemoglobin gets out there, it starts letting go of oxygen. Well, each hemoglobin has 4 oxygens to let go. So that's actually a pretty fair amount of oxygen. And hemoglobin has some things built into it that tell it primarily where do you want to bind oxygen. It has some amazing things built into it structurally that help it to do that. Well, let's look at one of those. It's called the Bohr Effect, B-O-H-R. The Bohr Effect is a phenomenon that was discovered by a scientist named Bohr many years ago. And in this Bohr Effect, it was found that if you take hemoglobin and you measure its oxygen binding, that's the percent saturation that we saw before, as a function of the oxygen concentration like we saw before. But in this case, instead of comparing hemoglobin to myoglobin, we compare hemoglobin at various pH values. We see something interesting happens. Okay? At the highest pH value, hemoglobin had the highest affinity. The curve is farthest to the left. As the pH value decreased, we see that hemoglobin has a lower affinity. It's giving up oxygen more readily. But what does that mean for the body? Okay? Well, rapidly metabolizing tissues, like let's say our muscles, for example, rapidly metabolizing tissues release something. They release protons. And we've seen earlier that the release of protons lowers the pH. And if we go from a high pH to a lower pH, we see that hemoglobin is losing affinity for oxygen. It's letting go of oxygen. All right? Well, that makes really good sense because what tissues need oxygen the most? Rapidly metabolizing tissues. So rapidly metabolizing tissues are sending out one signal. We'll see there are others, but one signal that says, hey, we could use some oxygen here. Those protons that are released bind to hemoglobin and cause hemoglobin to let go of some of its oxygen. That then feeds those rapidly metabolizing tissues and lets them do their thing. Another molecule that has effect on the binding of oxygen by hemoglobin is carbon dioxide. Now, carbon dioxide, I think most people know, is another thing that's released by rapidly metabolizing tissues because they're going through a lot of oxidation. If you measure the carbon dioxide concentration around rapidly metabolizing tissues, you'll see that it's higher compared to resting tissues. And what happens with that? Well, this graph compares 3 things. It compares hemoglobin at a pH of 7.4 with no carbon dioxide, compared to a pH of 7.2 with no carbon dioxide, compared to a pH of 7.2 with carbon dioxide. And if we look at that, we see that 7.2 has lowered affinity for oxygen compared to 7.4. That's the Bohr effect that we saw before. But if we compare 7.2 with no carbon dioxide to 7.2 with carbon dioxide, what do we see? We see again a reduced affinity for oxygen. So carbon dioxide binds to hemoglobin and favors the release of additional oxygen. And that's important again because rapidly metabolizing tissues are releasing carbon dioxide, and there's another flag that says, hey, we could use some oxygen right here. Well, this figure shows the phenomenon of cooperativity that I was describing earlier for hemoglobin. So let's go through it and understand a little bit better at the subunit level of hemoglobin what actually happens. If we look at hemoglobin on the left side of this figure, what do we see? We see that hemoglobin has 4 subunits, and each of those subunits does not have oxygen bound to it. The 4 subunits might, for example, have come from the tissues and they're headed back to the lungs. This would be what we call the T state, the tight state, where they don't want to bind oxygen. Now, in just a little bit, we're going to see one of the reasons why hemoglobin is in the T state at that point, but suffice it to say, it's in the T state. It doesn't want to bind oxygen. It gets into the lungs and the oxygen concentration is high. So when it's high, what happens? Well, one of the oxygens literally forces its way onto the first subunit. We can see that happening right here. And that first subunit has its configuration changed as a result of that slight movement of the iron atom that I talked about before. That changed that square schematically into a circle, and that circle now interacts with its partners differently than it did before. Those 2 partners that look like squares start to round their edges because they're being affected by the circle here, and as a consequence, they are becoming much more receptive to binding of oxygen. And so we see cooperativity happening. We see the first one changing the next 2. We see the next one moving along, favoring the binding of additional oxygens, and we see eventually that we get all the way over here to the side, and we've converted everything into 4 circles, each one of which has bound to an oxygen. So the binding of the first one influenced the binding of the others, and it influenced them favorably, causing that oxygen to bind. This model I've just described to you is something that I talk about in class called the sequential model. And it's sequential because it happens one at a time, and it's sequential because the binding of one changes the others, okay? There's a sequence that has to happen. The first one changes the second one, changes the third one, changes the fourth one, okay? That sequence turns out to be important, and we'll see later that there's another model that is important for enzymes that is different than what we see here for hemoglobin. Now, the last thing I want to say about signaling molecules that cells send out to hemoglobin is actually shown here. We can see on this figure to my left, we can see the overall schematic structure of hemoglobin. Hemoglobin has 4 subunits, 2 subunits called alpha and 2 subunits called beta. And they're arranged in a scheme like you see on the screen here. Notice that there's something called the hole of the donut, and the hole of the donut is the place where a molecule can very nicely fit in. This literally looks like a donut. And the molecule that can nicely fit into here fits into it just precisely, at least in adult hemoglobin. That molecule that fits into that structure, excuse me, is known as 2,3-bisphosphoglycerate, or 2,3-BPG. Its structure is on the screen. I don't ask you to memorize the structure, but I show it to you so that you know what it looks like. 2,3-BPG turns out to be a really interesting and important molecule. Now, we saw that rapidly metabolizing tissues released protons. They also release carbon dioxide, and both of those molecules can bind to hemoglobin and favor the release of oxygen. And 2,3-BPG is like them in that sense. It also is produced by rapidly metabolizing tissues and can bind to hemoglobin and favor the release of oxygen. So it's also signaling, hey, we need some oxygen here. 2,3-BPG has some other interesting considerations for it. So I'd like to just think about hemoglobin moving through the body and how that it would play its role. If we think about 2,3-BPG, it fits in that hole of the If we think about 2,3-BPG, it fits in the hole of that donut. And what 2,3-BPG does is it converts hemoglobin from being in the R state, which was the relaxed state that was binding oxygen, to being in the T state, the tense state, which doesn't like to bind oxygen. That is how 2,3-BPG is causing hemoglobin to literally squeeze out the oxygen so that the tissues can have it. all right? Well, 2,3-BPG is a molecule that's actually produced as a byproduct of glycolysis, which is the breakdown of sugar in our body. That is important because it can be used by the body for other purposes. So let's imagine that I've got hemoglobin that has gone from my lungs, it's gone to the muscles, and the muscles are producing a fair amount of 2,3-BPG. When they produce 2,3-BPG, it binds to the hole of the donut, hemoglobin changes into the T state, and when it changes into the T state, oxygen is released, okay? And now that hemoglobin begins its trip back to the lungs. Well, normally, 2,3-BPG does not stay in hemoglobin tightly, okay? It's not covalently bound. So it can be released by the hemoglobin on the way back to the lungs, and in most cases, it actually is. That means that as it goes back to the lungs, 2,3-BPG gets dropped off somewhere, and when it gets dropped off, the tissues that are there can grab it and actually use it for energy. Meanwhile, hemoglobin is left in the T state. So that cooperativity that I described before is really important because that first oxygen coming in there, binding to hemoglobin, causing the hemoglobin to change into the R state, the relaxed state, where it wants to bind oxygen, is really, really important, okay? Now, another consideration about 2,3-BPG that's important for our purposes is that 2,3-BPG is high in the blood of smokers. So because it's high in the blood of smokers, it means that if the hemoglobin lets go of a 2,3-BPG, it's more likely to pick up another one on the way back to the lungs. Well, what happens if 2,3-BPG is stuck to hemoglobin when it gets back to the lungs? Well, that's bad news because what happens is it'll hold, the 2,3-BPG will hold hemoglobin in the T state, and the hemoglobin will not want to bind oxygen. It'll have very low affinity for oxygen. That's what the T state does. Cooperativity won't kick in. Cooperativity can't help it. It's because of 2,3-BPG being high in the blood of smokers that smokers have reduced oxygen carrying capacity, and that's why they have trouble climbing stairs without getting out of breath, for example. Well, we can actually see the binding effects of 2,3-BPG on hemoglobin and also that of carbon dioxide, as you can see here. There are three lines here, the middle line of which is just normal hemoglobin. If we increase the pH, if we decrease the carbon dioxide and we decrease the 2,3-BPG, we get the curve that's on the left. If we, on the other hand, decrease the pH, we increase the carbon dioxide and we increase the 2,3-BPG, we see reduced affinity and we shift the curve to the right. Again, consistent with everything that I've been telling you so far. Now, there's another consideration for hemoglobin that's very interesting. So what I've been describing to you so far has been the hemoglobin of adults. And adults' hemoglobin is slightly different than the hemoglobin that's found in the fetus. The fetus in the womb of the mother it makes a slightly different form of hemoglobin. The hemoglobin of adults is called alpha 2 beta 2 because 2 of the protein subunits are known as alpha and 2 of the protein subunits are known as beta, and that's what adult hemoglobin is comprised of. Hemoglobin in the fetus, on the other hand, is slightly different. It's known as alpha 2 gamma 2. That is, the 2 beta units are replaced by slightly different units. They have hemes and everything, but slightly different units that are called gamma 2. The significant effect of that is that the gamma subunits are slightly different in structure, and so the donut hole that we saw before for the adult hemoglobin is not exactly shaped the same way. What does that mean? That means that 2,3-BPG can't bind in the donut hole of the fetal hemoglobin. Well, what did the donut hole binding of 2,3-BPG do? It caused it to flip into the T state, and it caused it to release oxygen. And that was important because adults are running around, adults are flexing muscles and needing very different concentrations of oxygen. On the other hand, the fetus doesn't have those very differing concentrations of oxygen. And it is more interested in binding oxygen because it has to take oxygen away from the mother. Because the donut hole is different in the fetal hemoglobin, 2,3-BPG cannot bind. And because it cannot bind, fetal hemoglobin is much less often in the T state than the adult hemoglobin is. Now, what does that mean? That means that fetal hemoglobin, therefore, has a greater affinity for oxygen than the adult hemoglobin does. So therefore, the fetal hemoglobin, which you see the adult compared to the fetus, the fetal hemoglobin can take oxygen away from the adult hemoglobin, and that's how the fetus gets oxygen from mom.